All right, we're going to move on to part 3, which will hopefully be pretty short. And we're going to cover 2 different topics in this part of the lecture. One is complementation analysis, and then the other is X-linked traits. So complementation analysis. So now we are moving to the question of whether individuals that have the same phenotype have the same genetic basis. So if I gave you if you're studying pea plants, and you have a dwarf pea plant, you've defined a gene called D, and you think you understand the genetic basis. And now let's say you go out and you have a friend at another farm, he's growing peas, and they say that they have also gotten a plant, a dwarf plant, and they're wondering if you want to study it. You say yes. And so one of the first questions you might ask is, are they dwarf for the same reason? Are they short plants for the same reason? Is it the same gene that's affected? Or perhaps they're completely different genes? And so this kind of thought motivates the purpose of complementation analysis. And it's become very common in the lab. So if you're a geneticist, if you want to go work for a lab that does genetics, you might work in a model organism like mice. You might work in a model organism like Drosophila. You might work in a model organism like C. elegans, which is a 1 millimeter long nematode worm. And you might do a genetic screen. So whatever you're interested in. If you're interested in behavior, you might screen for flies that have defects in a particular behavior. And the way you do that is you mutate the flies, and then you just screen through thousands to 10,000s of them looking for the phenotype that you're interested in. So maybe you're interested in flies that have better memory. Maybe you're interested in flies that live longer. No matter what, you're going to try to identify as many flies as you can that have that trait. And after you're done, you're going to have a large collection of these. And you want to study these. And so one of the first things you want to know is how many of these flies contain alleles in the same gene. And so how do you do that? So the way you do that is with complementation analysis, and then you define complementation groups. So these are groups of mutations. These are groups of mutants where all the mutations are present in the same gene. So what is the procedure for going about that? So let's start with an example of flies without wings. And so you are an experimenter. You have isolated 2 different flies, and neither one of them have wings. So this is your fly 1. Here's your first fly, and here's your second fly. And we're going to distinguish between 2 possible situations. So you have your 2 flies, and you want to know if they're in the same gene. And so the idea behind this analysis is that the wild-type phenotype is dominant, the mutant phenotype is recessive, which is kind of indicated by the names of the alleles that we used. And so in the first case, the mutations are in separate genes. So there's 2 different genes that control wings, and if you don't have functional copies of either of these genes, the flies are just never able to produce wings. And so this is shown here by kind of the location of the mutation. So this fly has a mutation in gene 1, so we're showing the mutations approximately at the beginning part of the gene, but everything is good with the second gene. And now in the second fly, gene 1 is okay, but gene 2 contains a homozygous recessive mutation in this gene. And so what happens when you cross it? So you cross these 2 together. So this individual produces 1 gamete, this produces 1 gamete. And so you produce a heterozygote individual that has the following genotype. So at the first genotype, this individual has this, and then at the second gene, this individual has the following genotype. And so this individual has 1 functional copy of both genes, and so it's actually able to produce wings just fine. So because it has functional copies of both genes, if we looked at the phenotype of these F1 flies, you would see nice wings. Now let's distinguish this to the case where both flies have distinct mutations in the same gene. And so we can see this fly carries a mutation in gene 1 and occurs somewhere in the start of the gene. This one has an independent mutation that's a little bit later in the gene. In both cases, it messes up the function of the gene, so it's unable to properly form a wing. So what happens if we cross these two flies together? So again, these are individuals that breed true, they're homozygous. So this can produce one gamete, this produces one gamete, and so all of the F1 individuals are going to be identical to each other. But now we see that the genotype is different. So in the first case, gene 1 carries 2 defective alleles, and gene 2 contains 2 wild-type alleles. So because gene 1 has defective alleles, it's unable to work properly, and the flies never actually produce wings. And so this is when we say no complementation occurs. So these mutants can't complement each other to allow for the production of wings. And so this is your strategy, this is your criteria. So if you cross 2 homozygous individuals together, and they're able to produce a wild-type phenotype, then you say that complementation occurs. And then you also know that you've identified 2 separate genes that you can study. In the reverse case, if you cross these flies together and you don't produce wings, then you know that these alleles fall in the same gene, and you have defined a complementation group. So a complementation group is basically a set of alleles that all cannot complement each other. So finally, we're going to cover what happens if the gene is on the X chromosome. So this violates the assumption that individuals have 2 copies of each gene, and that's because male individuals only have 1 copy of the X chromosome. And so it actually has a pretty dramatic change in the offspring ratios. So if we took a red fly and crossed it to a white-eyed fly, typically, assuming that they're breeding true, so let's say red is dominant to white. If this was an autosomal gene, and we cross these individuals together, we would expect all of the F1 individuals to have red eyes because they would all be heterozygotes. However, now when we cross this, it depends upon the sex of the parents. So we can do this cross in 2 ways. We can take red females and cross them to white-eyed males, or we can take white-eyed females and cross them to red-eyed males. And so now, in this first case, we kind of see, as expected, you have 100% of the animals have red eyes. But in the second type of cross, we see only 50% of the animals have red eyes, and 50% of the animals now have white eyes. And further, there's a sex component to this. So only the females have red eyes, and only the males have white eyes. Again, we can intercross these individuals together, and so in this case, we see a 3-to-1 ratio of red to white, which is kind of as expected. But now we see the sex difference arise as well. So 100% of the females have red eyes, and 50% of only 50% of the males have red eyes. If we do the cross in this direction, we no longer even see that 3-to-1 ratio. Instead, we see a 1-to-1 ratio. So 50% of the females have red, 50% have white, 50% of the males have red, 50% have white. And this can simply be explained by looking at sex-linked traits. So just thinking about how the X and Y chromosomes are inherited. So let's say we look at an individual with a trait of hemophilia. So hemophilia is a human trait that you can have that's linked to the X chromosome. So we cross an individual, a male individual that has hemophilia, to a female individual that is a carrier for hemophilia. And so we know this is the male individual because it has the Y chromosome, but this Y chromosome does not carry a copy of the gene that causes hemophilia. So this male individual only has 1 copy of this gene. And so you no longer even think about recessive and dominant relationships because those assume that you have some sort of diploid structure for a gene. And so in this case, you only have one copy. So what happens if we do the cross? So we have a female individual that is a carrier, and we have a female individual that is that has hemophilia. We have a male individual that does not have hemophilia, and we have a male individual that does have hemophilia. So the activity that is going to be part of class is to kind of look at this in much more detail, and at the end, hopefully you can understand how these crosses can arise, and that's kind of part of the point of doing the work from the activity. Finally, you can also have this in humans, and you can also do this sort of analysis on pedigrees. So previously we talked about distinguishing between recessive versus dominant traits or diseases in humans. Now we can also worry about whether or not a trait is X-linked. So is the trait linked to the X chromosome? An example of this is red-green color blindness. So males are much more common to have red-green color blindness than females. So probably some of you in the class will not even be able to see that there's a 15 actually within this group of dots, because you might be red-green colorblind. And so one of the ways that you can distinguish this is you look for rules where you know if the gene is X-linked, then certain things must happen. And so one important rule for that is that if you ever have a female that has the trait, so in this case we have a female that is colorblind, they can only produce sons that are also colorblind. And so why is that? So we know that this individual is not colorblind, so this male individual must have an X chromosome. So let's see the offspring that they've had. So they've had 4 different offsprings. They've had a male, a female, a male, and a female individual. And we notice that the 2 males have color blindness. And so why is that the case? So if we look at what was inherited from this individual to this individual, we know the male must have passed on the Y chromosome to this individual. Because if the male did not pass on the Y chromosome, then this individual would be female. The mother can only pass on one of these two, can only pass on one gamete, because she is homozygous for the colorblind allele. And so all of the male individuals will have to be colorblind. And further, if we look at all the females, they must have received a copy of the X chromosome from the father that does not carry the color blindness allele. And so all of the females must not have the trait. So this is kind of the logic, it's almost like a puzzle, right? It's like a little logic puzzle that you're doing, Sudoku or something like that. So that's the kind of like logic puzzle that you would take in order to try to figure out is something X-linked or is it autosomal. All right, so hopefully you enjoyed that chapter. This is now getting into some more interesting type of genetics. And now we're going to move on to mapping for the next chapter.